Hello, folks. My name is Dane Banks, and for Protein Project 3, I'm doing ferredoxin. Ferredoxin is a very simple protein. It has generally one purpose, and that is to transfer electrons. Ferredoxin has four cysteines connected to four sulfurs connected to two irons, which are interconnected by two sulfurs. So the cysteines don't really matter for the overall structure, but they're there. From there, the sulfurs and the irons are the primary focus of its function and how it works. So in the very center here, you'll see two sulfurs. These are very important. In electron transport for this protein, those two sulfurs are the ones that receive extra electrons. And then from there is stabilized by the four surrounding sulfurs out here. So, the overall structure here is very reminiscent of a red blood cell. Whereas red blood cells use oxygen to transport, are used to transport oxygen. For this, the electrons directly attach to the sulfurs in the very center and are stabilized by the surrounding sulfurs. So now that you kind of see how the overall structure is and how it will relate, we're going to jump over to a small little thing right here. So ferredoxin has a bunch of options here. Ferredoxin is found throughout mitochondria, chloroplasts, as well as in bacteria and a bunch of other cells. So it is very important to look at what part of both the cell and what type of cell that ferredoxin is in. For this example, I have ferredoxin in a chloroplast, specifically during photosynthesis. Though you can find them in other places. So overall, the reaction for ferredoxin in this example has two reduced ferredoxin in solution with NADP plus and H plus ions. From there, those are turned into two ferredoxin, which have been oxidized, and NADPH, which we all know is really a really good transporter. So how does this function work? Well, it's split up into three different subreactions. So the overall reaction is ferredoxin, which has been reduced. It's been already received the electrons, reacts with ferredoxin NADP plus reductase, which has been oxidized in this case, and it turns into a ferredoxin that is oxidized, and FNR, which is what ferredoxin NAD plus reductase, steals one of those electrons. From there, it goes back in for a second round and steals the last, a second set of electrons, and turns into ferredoxin NADP plus reductase, which has been reduced. From there, the FNR, which has been reduced, reacts with NADP plus to turn into FNR oxidized and NADPH. So overall, the function basically goes as follows. Ferredoxin, which has extra electrons, there's two of those. Both of those are stolen by ferredoxin NAD plus reductase and given to NADPH, making it neutral and then negative, allowing it to attack and receive a H plus ion. So that's one of its many functions. And as such, you can see this has very large versatility. It can go all over. It can be in mitochondria, it can be in chloroplasts, it can be in other bacteria, it can be throughout many of the ecosystem and many structures. Ferredoxin is found, or in this case, in these researches, is the bacteria known as Clostridium pasternanum. The SMR code is P00195, so that will help you if you want to learn more about it. That's what ferredoxin is. Its basic function is electron transport, and as you can see, it's fairly simple. I hope you all have a great day, and I'll see you in class.